Thank you. So Christie Bahlai, I'm gonna let her talk. She does a lot of different things. And so I think I'll just let her talk. But thank you for being here, Christie. We really appreciate it.
Well, thank you so much for asking me to come, Gina. So I'm Christie Bahlai, and I'm an assistant professor at Kent State University. I'm about, I guess I'm in my 4th year. Just, time is weird right now. And so trying to keep track of what my time, how long I've been at things, and oh my. But today I'm going to talk a little bit about time and conceptualizing time through data through the lens of a really specific problem. Now, if you don't like insects, you're going to love them by the end of this talk. But I am an insect ecologist by training and a data scientist sort of by accident. I became a data scientist because when you're working with insect data, there's a lot of it, and you start to recognize that there's a lot of issues that you have to learn about when you're trying to really authentically model a system. And so I'm going to be talking to you about a phenomenon that's been really popularized over the last probably about 3 or 4 years, which is the insect decline phenomenon. Now, I'm sure you've probably seen headlines about the insect apocalypse, right? Or a few of you have. A few of you went, well, what's wrong with that? That would be okay if the insects went away. It wouldn't. It would absolutely not be okay if the insects went away. And so I've been working in insect dynamics for also a longer time than I'd like to admit. But I've been thinking about population patterns and how we model them and why our tools are inadequate for answering a lot of the problems. Okay, so let's see, is this gonna, there we go. So when I talk to insect ecologists, I have been told that I am a very depressing person. So when you're the quantitative person in the room, I'm sure many of you have had this experience. You're always the person who is telling someone that no, their idea can't be done, or no, the tool doesn't say that, or just really being the bummer. And unfortunately, when I gave this very similar version of this talk at Ohio State to the entomology department, I was told that this was the most depressing talk that they had ever seen. So I've tried to, make it a little bit happier, but as quantitative people, I think that you'll relate a little bit more. So I mentioned the insect apocalypse as this sort of driving factor. So this was a article in New York Times Magazine, late 2018, based on a paper that had come out in PNAS, basically claiming that insect life was in uniform, terrible decline across the planet, in addition to several other studies that had just sort of said, well, in this more localized place, we're losing insect life. And when people make really bold scientific claims, you have the pedants in the field coming out of the woodwork going, well, the data don't really say that, or my data don't really say that. And so this was one of those cases. So we have this New York Times piece, and then this is one of the papers that was supporting the New York Times piece. Essentially, what they found in this particular paper, the Lister and Garcia paper, is that arthropod abundance was dramatically going down. It was related to climate change, and this is based on data from the Luquillo rainforest at the long-term research site in Puerto Rico. So what they'd done was they'd conducted surveys in the 70s, gone back and conducted surveys again, and what they found was some pretty dramatic changes. Okay, so here's some of their data. You can look at these and go, whoa, those are some different numbers. The authors looked at this and said, well, in 1976, there was 473 beetles or bugs in that trap. And in 2012, there was 13. So 13 is smaller than 473. And so there is definitely a climate-driven signal. Now, then the scientific controversy started. And so you can hear the tone if you read this title aloud. So I'm just gonna read it aloud for you. Populations are not declining and food webs are not collapsing at the Lakio experimental forest. So this is a paper that was done by the people who were running the site. So I later talked to some of the authors of this particular rejoinder. And so Tim Showalter, who's in the middle there, a colleague of mine, we always run into each other at conferences, and he said, oh, that paper. The first thing we did when we saw that paper come out is we said, how did we miss this in our data? How did we miss this pattern if it was this strong? Why didn't we see it? And these guys came in and found this really strong trend. And then he said, we looked at the covariates and we said, well, of course the canopy traps are catching fewer insects. There had been hurricanes and the canopy wasn't there anymore. So the context in how this data was interpreted is everything. And they went on and they matched it to climate patterns. And so they dissected the central claim of the original paper and said, in fact, we actually see a decline in the temperature at some of our sites. So we actually see an opposite effect of climate than you would have actually seen if you're accounting for all these other covariates. So context really matters in insect dynamics and a lot of things. And so this leads us to a question of, well, why can't we figure this out? Why are insect dynamics so hard to study? And so we're going to do a thought experiment based on the work of Lister and Garcia. So like good quantitative ecologists will create a nice blank figure. And we'll use a nice quantitative scale of few to many. And then we will use a temporal scale of then to now. And so we'll go back and we will do our surveys in the past, and we'll find that there's a certain mean value of insects when we capture, and we repeat the survey several times so we can put an error bar on it. And then we go back at a later point and we repeat the survey, and we do a few subsamples. We can have some error bars, and then we put this into an ANOVA, and we find that there is a statistically significant difference. And I'm going to tell you right now that as an insect ecologist, this is largely where my statistical training left off, my formal statistical training. And so from this, we'd say, well, there are fewer insects now than there were then. It is a statistically significant conclusion. Well, insects are declining. Sorry, there we go. So we can be a little bit more precise. If we wanted to be able to infer a decline process, we're going to have to do a little bit more than that. So we're going to have to give a scale that we're measuring the number of insects on. And we'll probably want to actually measure what our time points are. And from there, we could do a linear regression. Ooh, yeah. And oh, it's highly significant. And from there, we sort of feel comfortable doing things like this, where we're extrapolating from the relationship. And this is actually the type of analysis that several of these papers actually use. This is their math. They essentially drew a line and followed the line to the axis. And they concluded that insects would be extinct by the point where the line meets the x-axis. And so that's a little problematic. But the thing is, we know, because we've worked in natural systems, that the data looks a lot more like what you're seeing here. So we've had these snapshots that we use, and we've got this really, highly significant p-value, but we're losing all the nuance. And so we could even do a linear regression on these data and get the exact same slope and get the exact same significance. But we would feel a little bit less confident if we're considering the whole context of the data set when we're making that extrapolation. The way we present the data has a really strong effect on our confidence in our conclusions and our interpretations of our conclusions sort of in context. And that's the problem with populations. Populations are really messy, and we're trained to use our statistics to really compress them down into very simple messages. And that's kind of messy, and that's why we have all of this disagreement. So we come back to the question of why are insect dynamics so hard to study? Population dynamics are both an instantaneous measurement that we take at a time, but also a superimposition of multiple processes that they take to get to that point. So assuming the snapshot is the whole truth is losing so much of the nuance. And so sometimes how we get to a point matters as much or even more than what that point is. And so this question sort of became a big focus for me a few years back. So I'd been doing some work with some students on dynamics of fireflies at Kellogg Biological Station. So this is actually kind of a funny story. We were using a data set resulting from the long-term ecological research site there, where the project had changed hands in 2004, and a technician added a column to a data sheet for fireflies. So insects were, mostly lady beetles were being surveyed for, and they incorporated a column to add fireflies. And so I became a postdoc on this project. And I was looking for a data set to work with for a quantitative class that I was teaching. And I mentioned to my PI, hey, could I use the firefly data? I don't think anyone's done anything with it. And he said, what firefly data? As it turns out, this is the longest systematic study of fireflies known to science. We've got more than 12 years of data now, but at this point we had 12 years of data systematically collected from a randomized complete block design, plant community structure every year, just beautiful data. And no one knew it existed. It was just bycatch on this main experiment. So in this particular data set, we had this really rich set of observations. We had all of the contextual information. It was best case scenario for doing a project with students. And so students came up with a question of essentially just modeling when firefly activity occurred. So the phenology and sort of looking at when we would expect firefly activity peaks at the site. And we wrote a paper, we wrote a little preprint up. We were actually able to disentangle temperature and precipitation, which is something you can barely do unless you have a lot of data. And so it was just this beautiful little thing. And we got a little bit of attention from the media when the paper went out. Was interviewed by Science Magazine, and I was just so proud. I was really pleased with myself because, you know, we had this wonderful phenology model that really was predicting activity very, very nicely. And the interviewer kept coming back to, yeah, but are fireflies in decline? And well, I don't really know. So here is the firefly data. Each plant community, so we had 10 different plant communities from 2004 to 2015 it was. You can see that there's a pattern here. So when the reporter asked me, so are fireflies in decline? I said, kind of being a little bit of a smartass, I'll admit. Well, I don't know, but if I had less data, I'd give you a much surer answer. Because as you can see, there are periods of decline, there are periods of increase, and in fact, it's probably a cyclical dynamic, which is not unexpected for an insect population. But this got me really thinking about the question of trajectory. This is a really fundamental thing that we want to know from our systems. Where are they going? And with 12 years of data, I was unable to answer that question. And in fact, it made me really uncomfortable, because if we had had less data, I probably would have given an answer, right? And so this got me really thinking about this concept of how when we look affects our answer so dramatically, and how long we look. So this is right here is data from the Peter Doran paper from Nature from 2002. This is one of the most famously cited by climate change deniers paper that we have. So this is the Antarctic climate cooling paper. This is where they found a significant decrease in the temperature in the McMurdo dry valleys in Antarctica over a period of, I think it was 17 or 18 years, or in the 80s to late 90s. And you can see this is the data here. Okay, will my mouse show up? Yes. Okay, so you can see that they started their data collection in a year where there was an abnormally high temperature, and they had an outlier. And so that actually causes the linear regression to be tipped. And if you know how linear regressions work, they're kind of like teeter-totters. And when you have an outlying point near the end or the beginning of the teeter-totter, it puts more weight on the tip of the slope. And so what you have is a situation that, unless there was several additional record high years later on, you're basically creating a situation where it's impossible for the slope to be anything but a negative slope when you start with an outlier. And that's kind of bad luck, really, isn't it? Like, they just happened to start during that time period, and they never had another outlier year quite that high. And so during that time period, they ended up with a negative slope. In reality, there's probably not a really strong trend in either direction here in these data, but it becomes this situation where this statistical result, it has a lot of weight put on it in the media, in the popular culture. There's also the issue of natural variability. So I was telling my Firefly story to a colleague at Kellogg Biological Station at the time. And he said, "Oh, you think that's bad?" I had a paper. This paper he eventually published in Nature, but he said it just had the worst time in review because my p-values were bad. He said, so there was a reviewer that I'm showing this trend in this paper, and there was a reviewer that just kept coming back and saying, well, these studies, all these other studies, found so much stronger results than you, or contradictory results to you. And you have these really, really just barely significant p-values. And so you have no statistical confidence in your result. And he's like, well, we have 20, 30 years of data. Why is this an issue? And so there was just this big resistance on the part of the reviewers to interpret things with a little bit more nuance, because the statistical tools that they were using were sort of valorized, because we had this idea that, we have a significant relationship. I mean, if he had done this study from 1998 to 2001, there would have been a really strong trend there and a really highly significant p-value. And, it would have been a really tight result, a good result. And so this reviewer really valorized that sort of thinking. And then there's issues potentially of site selection bias. So this is kind of going back to our Antarctic data, but more on a population level. So this is a figure from easily one of my favorite papers from last year, a paper by Ariel Fernier, Easton White, and Stephen Hurd. And so this paper started with a thought experiment on Steve's blog. He said, I'm going to posit that all populations under monitoring are in decline, and here is why. If you go out to the field to monitor frogs and you have 3 ponds that you can monitor frogs at, you're going to choose the pond with the most frogs at it because it's the best site to monitor frogs at. But you are probably capturing that at a high point in its population cycle. And so the choice of your site is potentially biasing the pattern that you see. And so they demonstrated it with this. So imagine you go and you collect your data at your frog site. And then you have what looks like a pretty natural population dynamic. This is pretty classical population data that you'd see. If you started at a good site, probabilistically, there's really low odds of the time you go back and take your snapshot of landing at that same high point ever again. And so they actually conducted a whole bunch of simulations of these data and found that probabilistically, when you're selecting sites that from a set of sites that have a variety of different dynamics, you're likely to choose one and very likely to end up with data that looks like a decline, even if it's sort of a classical dynamic like this. It's very unlikely that you would choose the pond with no frogs, because that seems like a bad place to go monitor frogs. And so having that context and having sort of a historical understanding of the site can help deal with that potential bias. And then while we're talking about biases, so this is another one of the insect decline studies. And the title of it is Worldwide Decline of the Entomofauna, a review of its drivers. And I am not a geographer, but what do you notice about this map? The use of worldwide seems a little bit pushy. And so, in fact, here, this part of the map is probably the part of the world that has the most insect diversity. And you can see very little data from that part. This study also had some other problems in that when they conducted their meta-analysis, they searched the literature for insect survey decline. They had the word decline in the survey. And then they put all the data together and they found that insects declined when you search for insect decline, as it turns out. So there's more than a few issues with this study. And so here I am pointing out all these problems, but what it comes down to is insect dynamics are really hard to study because it matters how we measure, it matters when we measure, it matters how often we measure, when we start, and for how long. It matters how much inherent variability our measurements are capturing, and it matters what else is happening when we measure. So, you know, you're not necessarily catching a natural dynamic if you've got some sort of external influence on it. And then, of course, it also matters where we measure. But like I said, I'm not a geographer, so I'm a temporal ecologist, so we'll ignore space for now. But we have some very human constraints on data collection. So this is a plot of study. Okay, so I did a quick lit search for length of ecological studies. So the solid blue bars are the last 15 years and the shaded blue bars are the previous 15 years. And so what this is I did a search for ecology blank year study. And for each of those, I just subbed in a different number. And so what we see is that, well, first of all, there's more studies in the last 15 years than the previous 15 years. But by and large, most ecological studies are in the range of under 5 years in length. We do have some longer ones, but they tend to peak out around 2 to 3 years. So why is that? Well, grad students, funding, lots of reasons like that. And you know, this is a human constraint, and I don't see us being able to completely resolve it anytime soon. But I became really interested in this as a problem. I became interested in asking the question of how we watch the system ultimately affect our conclusions about the system. And so like, what's the observation period's effect on our outcome? And what's the length of the observation period? And can we separate these trends in ecological systems from the natural variability and the underlying processes? And so this is where I got into tool building. So this is actually where I started actually replacing myself with computers, or replacing grad students, but myself as sort of the hypothetical grad student in this situation. So the broken window experiment was born. So the idea is we're looking through our individual windows, and our individual windows are our biases, our position in life, just when we happen to be out in the field. And we have those things, but we aren't necessarily conscious of what the panes of our window are. And I said, well, we've got long-term data. Why don't we just simulate someone doing this? And so what you have here are red rectangles to indicate my time as a master's student and my time as a PhD student. And so you can see that I would have made the conclusion that specifically fireflies were in decline during my master's. And then I would have done an intervention, come back during my PhD, and then said, oh, my intervention is working. Look, I've saved the fireflies, with the numbers going up like that. So this is objectively terrible, right? This is an objectively terrible way to do data. But if you do it enough times, it becomes statistically significant, right? I created an algorithm that we call the bad breakup algorithm to create broken windows. And within each window that we look through, measure the strength of the relationship, the variability of the relationship, a decision rule about the relationship, and our confidence in the decision. And so here's our path to enlightenment. We get our raw data and we come up with some sort of summary metric. So number of fireflies per year, or number of fireflies per trap, or what have you. And then we feed it into the bad breakup algorithm. And then we compile our statistics. So we do our linear regressions where we do a slope, we get the slope standard error, the p-value, we can get the r-squared, basically everything that your standard regression is going to give you, and the thing that you would base your conclusions on if you had only that window. Then you do your statistics on your statistics, and you get your fundamental answers. Okay, so we're somewhere in between the statistics and fundamental answer step at this point, but you can see the enlightenment is coming. And so when you do this exercise with the firefly data, where we look at the data in no-till plots, what we see is if we just do a linear regression, and we can say, yeah, a linear regression is probably not what we'd want to do looking at those data. But if we did do a linear regression, what we'd see is a slight negative, non-significant slope through that data. And so we'll call that the truth. But if we did this for every possible interval of 3 years, what we'd find is we get a whole bunch of different answers. And so all the red dashed lines are statistically significant results and possible Nature papers. And all of the dotted gray lines are, oh, well, I'm putting my data in my file drawer. And so if you do this again and again, you can create a nice little animated GIF where you can see that the longer the window you use, the more likely you are to have a slope near your true slope. And then you can take all those slopes and plot it. And so this is what we call the pyramid plot. So we developed this, my group developed this as a way of visualizing it. And so what we do here is this provides sort of a quick visualization of, do we have enough data? So what we have here is the true slope in the center here, and that's the dashed line. And then every point on this plot represents the slope and the number of years that it took to get to that slope for all the different samples. And so you can see that when you have a 3-year data set, you have a large number of different slopes that can occur. And the size of the circle, and the circles are significant results, represent the R squared. So those are scaled by the R squared. So every time you have a big circle, you have a highly confident trend that's going on in your results. And every time you have a little circle, it's a less confident trend. And then when you have a red mark here and a little red X, that is a non-significant trend. And so you can see the longest time series gives us a non-significant trend. And we get trends up to, okay, so that's 7, that's 6. And so at 6 years, we have all of our slopes within 1 standard deviation of our true slope. And so we say, okay, well, reasonably confident that, you know, this is a reasonable answer when we're looking at that time series. But we also can strictly quantify that about 67% of our subsamples are giving us really misleading results before that point where we're seeing that stable trend. And so this is all scaled by the z-score, so it's all in standard deviations. And so you can see that we've got a pretty wide range of potential outputs. And another thing that we can see is that, you know, we could be wrong at any time. We can go to the field and collect misleading data at any time. But the problem that we really face is our confidence in it when we have this short time series. And so this is the blue line is our proportion wrong. So these are results that are statistically different from our true result. So the slope of our longest trend. And then this dashed line is the average R squared of those values. And so you can see that we've got some pretty high R squareds down at the low windows and then really low R squared values at the high number windows. And so what we see is, yeah, we could be wrong, but we're most likely to take it to the presses with a shorter window. And so we've actually taken this and used this on some long-term data. So working with Nick Haddad and Sarah Cusser at Michigan State, they took the algorithm and applied it to treatment differences between no-till and conventional till agriculture. And so in that system, they're really prone to having really dramatically different results in short-term trials. Basically, this technology has been around for decades and decades, and there are still debates going on about whether it's valuable or not. And so what they did was they applied this to the 30-year time series collected at Michigan State. And what they found was about a third of the time, you're going to have misleading trends in shorter term studies. And but after 10 years, no-till agriculture is almost always beneficial from sustainability standpoints, from the economic standpoints. Essentially, it's a good investment. It's just environmental variability masks that a lot of the time. So I had come up with this idea and was talking about it at a conference and found out that I'd gotten an NSF grant associated with it to build this algorithm. And I went to a talk by Easton White and realized, uh-oh, we are Darwin and Wallacing this. We had come to a very similar idea at about the same time. He had just published a paper, I had just gotten a grant, and instead of creating a lifetime of vitriol, we decided we would work together. And so we created, we recently wrote a commentary about how these resampling techniques that we've developed can improve environmental monitoring programs by helping us direct resources to the types of data that we need in order to be able to see meaningful trends in different population systems. And so that is currently in typesetting at Frontiers in Ecology and Ecology and Evolution, that one. So expect to see that one out soon. So this is all me telling you all of the ways that we can be wrong in ecological systems. And I suppose this is why I depressed the entomologists at OSU. But I want to sort of build on a little bit more of an optimistic note. So the way we collect data is in fact changing how we're doing ecology. And in fact, we're developing a lot more data that we are collecting passively. And so by passively, I mean observation networks rather than data that's produced as part of a manipulative experiment. And so this requires us to get cozy with inductive reasoning rather than in deductive reasoning. So we're not necessarily creating a controlled system. What we're doing is we're collecting a lot of data and collecting evidence and bringing it together in a different way. Now, data science has been very bold and made a lot of claims about how the data deluge would make the scientific method obsolete. Now, I like to cite this article because it came out in Wired magazine in 2008. And as you can see, the scientific method isn't dead yet. So maybe a little bit premature on that prediction, but there is a lot of really useful stuff that we can take out of data science as a complement for more classical methods. And so before I get in trouble, correlation is not causation. Just putting that out there. But this is also where I get myself in trouble, but it provides a hint. It is a really important way to provide hints about the system. And so we only have to go to examples from epidemiology where we can really dive into this. Now, it would be really, really unethical to prove once and for all that smoking causes cancer, right? Because we can't go and assign a group of children to smoke and then another group of children that we've randomized to not smoke. But we can do more observational-based epidemiological studies where we're attempting to control using sort of natural population, variability, but it does require a sort of changing of how we're looking at our reasoning. And because of this inflexibility, the tobacco lobby was able to really push back on increasing evidence about smoking causing cancer for years and years, because people weren't able to say, well, yeah, I did a controlled study and no, there were definitely more kids that got cancer in the group that we made smoke. And so the Bradford Hill criteria were actually born out of this need of exploring inference based on inductive reasoning rather than deductive reasoning. In situations where we can't do our randomized complete block experiment, we have tools for looking at these data and actually getting at what's going on in a system. And I won't go into them in detail, but they have really important guidelines that I think are very important for data science moving forward. Like we're all about correlations in data science. And so we have to really critically evaluate how the data are structured in order to really get at causation. So temporality as an example. So are you getting cancer after smoking, or are you getting cancer and then starting smoking? Like, is there evidence of temporality in the relationship? Gradient, does more smoking cause more cancer? So on and so forth. And does it complement experimental evidence? So these are the really important factors that we really need to consider as data scientists when we're inferring causation. And so these criteria have actually been applied to ecological systems. I used to call this a recent study because I was very excited when I found it, but it was from 2011. But so what this was examining the link between neonicotinoid pesticides and honeybee decline. And so what they found in this particular study was there is a lot of evidence that does associate it, but a lot of it doesn't actually hold up to the Bradford Hill criteria. And so the system, the system is probably more complex than sort of a one cause, one effect response. And so they actually developed some more guidelines in this paper about what other things need to be investigated in order to pull apart this relationship. But we're going to talk about lady beetles because lady beetles are my favorite. I mentioned that this Firefly data set came out of a lady beetle survey. So I worked with the long-term ecological networks lady beetle population data out of Kellogg Biological Station for my postdoc. And the year I had arrived, my then supervisor, Doug Landis, had published a paper where he said, you know, the long-term dynamics associated with lady beetles are just sort of beyond what we would have predicted. And we wouldn't have been able to capture this really interesting pattern had we not been studying it for this long. And so what he pointed out was there was sort of this classic invasion curve when a new lady beetle came into the system. And at some time point, its prey item, a primary prey item arrived in the system, which fostered this huge explosive dynamic. And then something else happened and it flattened out a little bit more again. And so I thought this was a really interesting problem. And so I started fitting models to it, as a quantitative ecologist would do. So it really looked like a density dependent model. And so I started playing around with some different models. And as it turns out, the classic stock recruitment curve, the Ricker curve, fits lady beetle population dynamics really well. And so I got really excited because I had a model that fit my data. Yay. But I got to sort of wondering, it's like, well, the constants within a model are both measures of what's going on at that time, but they're also reflections of the environment. Those constants in the model, so your N, your K, the K and the R, so K being the carrying capacity and R being the intrinsic rate of increase, are affected by the environment that they're in. So what if those constants could be used as environmental signals? And how would we determine that there's a change in those? Well, we could just throw different models at different times and see how R and K change. But I was unhappy with how biased that really felt. And so it's an approach very similar to the broken window approach. What I did was I developed a model selection tool that allowed it to allowed R to go in and refit the model at every time point combination. And then I gave it some decision rules about what a good fit represented, and then gave me an unbiased collection of the fit statistics and ranked the models for me. And so it just goes and it breaks the model, breaks the data set at every time, and it fits it. And so what the model ultimately told me was there were slightly different change points than what had been suggested by Doug Landis's initial paper. Specifically, there was change points in the time series happening. Oh, did I just unshare? It's still good? Okay. The share menu went away suddenly. So there is a break in the dynamic here. And so if you replot the population density in year one versus a population density in the next year, you get these really characteristic curves. And so what we see at this time point is this big shift associated with an increase in carrying capacity and an increase in intrinsic rate of increase. But then something happens and it returns to the previous dynamic, pretty much statistically identical to the previous dynamic. And so what we find here is this is the introduction of neonicotinoid insecticides in Michigan. The change in the dynamic of the higher trophic level lady beetle is affected by the control of its prey, to the point where it looks like from their population dynamics, the prey isn't even there anymore. And so this, I thought, was a pretty useful tool, but it's really correlative. But we got to try it in a few other situations. So what I did was I created a more generalized regime shift detector tool. and developed a means of estimating the confidence in the breakpoints. So what I did was essentially use a multi-model inference approach, which calculates the model weights, but associated with the breakpoints. And that gives us a relative ranking of how important different breakpoint combinations are. And so I applied this to monarch population data. So using their overwintering population densities at their sites in Mexico. And what we find is, well, first of all, pretty much a systematic loss of carrying capacity. That seems to be the biggest signal on this. Like, we're just losing carrying capacity, either in the overwintering grounds or in their breeding grounds, but that's what the signal is. And what we see is these change points. And so we see two strong change points, but one sort of weakly favored one. And so the weakly favored one isn't there. It is there only if these ones aren't. These actually line up with really strong increases in the uptake of glyphosate in Midwestern soybean here in 2003, and then in Midwestern corn in 2008. And so we see a signal that's probably associated with increasing weed control in croplands, which actually interestingly complements some work that's been done in Doug Landis's lab since that time. So he had a student who's recently finished, Andrew Myers, now Dr. Andrew Myers, I'll give him that, where he found that there might actually be a preference in monarchs for laying their eggs on milkweed that's occurring within field crops rather than within prairies. And so it looks like we might have selected monarchs that really like laying eggs in the landscape that we have created for them, and now are better controlling the weeds in. And so that's what the potential signal is. Now, admittedly, this is a hypothesis generating rather than a hypothesis testing exercise, but it is adding to the strength of evidence associated with multiple studies that are ongoing in these landscapes. And so, I mean, only because I was working so closely with Andrew and Doug that we were able to sort of pull together those aspects of the story. And it all comes together to make sort of a plausible explanation that we can go on and make more manipulative tests of in the future. So I want to end with how important it is to trust our data, but also being careful about creating the lore around the data. Sometimes you can't tell if data is being biased by lore. If you are a Star Trek fan, there are many episodes where data has in fact been manipulated by lore, and it has been influenced by its context. And so I just wanted to leave on that message. And then a shout out to my collaborators, and I'm happy to take questions.
Well, thank you, that was awesome. Questions from anyone in the gang?
Yeah, Christie, in the agronomy seminar, like 2 weeks ago, I think we had someone, I forget his name, but he talked about the monarch pattern. And he talked about exactly what you mentioned, the milkweed in the crop fields. We control it because we use glyphosate. And so that was linked to, he didn't go into the details though. So I'm really happy. And I remember being, so.
Well, so one of the really interesting mechanisms that seems to be coming out of a lot of the monarch researchers is that maybe this is a selection for these monarchs that prefer to lay in field crops, because, well, field crops tend to have fewer natural enemies in them. And so it's actually a safer place for your babies, at least until, you know, the weeds are absolutely controlled. And so you actually have a better survivorship of monarch larvae and eggs, potentially in these relatively uniform plant communities compared to sort of the more natural systems.
Yeah, he had also talked about the fact that a lot of the milkweeds are shade grown, and so they have thinner leaves and they're higher nitrogen content. And so that's really preferable for the little baby monarch.
Yeah.
So neat to see this all come together.
Yeah, some recent work by Doug and his postdoc, Nate Hahn, actually found that the monarchs do better on milkweed in under disturbance regimes. So like milkweed that's been mowed and then resprouts, they just love that stuff.
Right.
There's a hand raised from Audrey.
Yeah, hi, this is Audrey McCombs. Thank you very much, Christy, for a great talk. This is really interesting. I appreciated especially how you touched on some of the larger questions in science and how science is done these days in terms of how we bias, you know, how site selection can be biased and temporal selection can be biased. I very much appreciated that. But one of the things that I wanted to ask you, and I have a lot of sympathy for that. I'm a co-major PhD in ecology and statistics. And I started in the ecology department and moved over to the statistics department because I have a pretty good appreciation of how some of these statistical issues can be very relevant to the conclusions we draw in science. And so, like I said, I'm very sympathetic to where you're coming from, but I wanted to raise a question. Could you put up the slide of the Firefly data really quickly?
Yeah, sure.
And I have a point that I want to make and a question to ask.
Okay, let me pull back into that. Oh, sorry, it's lagging on me.
So while you're doing that, while you're pulling it up, you know, when I looked at that Firefly data, the first thing that I saw, which is something that you mentioned, was there seems to be some sort of periodicity or seasonality to that data. Yeah, that's what you can just leave it there, that's fine. And so, you know, the first thing that I thought of, sort of putting my statistics hat on, is, well, this is time series data. And so maybe one of the things that we could do would be to remove the periodicity and then look for a trend, right? It's a fairly standard time series analysis kind of thing to do. And so the point that I'm trying to make related to what you were saying about sort of the tools that we get when we're in graduate school about statistics pretty much end at regression. And I think that that's absolutely right. But we have more tools in our toolbox than just regression. And this data set, I think, is a really good example of where regression might not be appropriate. The correct tool to use here would probably be some sort of time series analysis where you're removing the seasonality and then, like I said, looking for trend. So the point is that I think we do need to expand our toolbox of statistical tools beyond just regression. But then the question I have for you is, it sounds like you work a lot with temporal data sets. Why didn't you do a time series analysis on this data when you got it?
Well, so at that point, we were mostly looking at within season phenology, because we looked at these data and we went, oh, there's 2 cycles. So that means that essentially we have 2 data points when we're fitting a time series. And so we weren't really comfortable making any sort of extrapolations at that point. And so it was in the context of teaching a class, and we just sort of followed up with our phenological models. But it was really interesting. The reporters was really drawn to that conclusion. And, you know, I didn't have an answer prepared because, you know, I sort of said, well, data quality, and sort of left it at that. I am looking forward to actually going back to this and looking at some sort of model that accounts for the periodicity, because it's really clearly there. But at that time, it was sort of just, that was the need was to answer that question. And it just got us sort of thinking, well, how often are people making that conclusion based on sort of that inadequate scale? Like, so this is a scale question, of course. We're asking, we need to be able to sort of look at things with a good fit from that periodicity. But we don't really have enough to be able to say that because we've only got the 2 cycles. And so we can't really draw the conclusions, but intellectually we're like, but you have 12 years of data. Why isn't that enough?
Okay, thank you. Well, I'll be interested to hear, if you do fit a time series to this, sort of what happens. But
Yeah.
Thank you very much. Thanks.
Yeah, it's past 1 and I don't want to eat up more of Christie's day. But so thank you very much for presenting. This was fantastic, Christie. And I'll send you the YouTube link to see your work.
Okay, great. Thank you so much for having me.
Yeah.
And if anyone needs to reach out, I'm pretty easy to find on Twitter, email, everywhere. I'm Googleable.
Definitely Googleable. Great.
All right.
Thank you
Thanks
so
so
much,
much.
Christie. It was
Yeah,
great.
Bye.